Hey guys, in this video we're going to go over cell division, which is unit 1.6 of the current IB biology syllabus. So first we're going to go over the cell cycle, which is the sequence of events between one cell division and the next. There's two main phases of the cell cycle, which is interphase and cell division. Interphase has three components to it: DNA replication, protein synthesis, and cell respiration. These are all functions that occur during interphase. And in cell division, mitosis and cytokinesis occur. Mitosis is the separation of nuclei, and cytokinesis is the separation of cytoplasms. Make sure you guys know the difference between the two. The following diagram on the right is a diagram of the cell cycle. You guys will need to know how to draw this, including that interphase have a G1 phase, an S phase, and a G2 phase, and that cell division has both mitosis and cytokinesis in it. Most cells follow the cell cycle, with the exception of cells that don't divide, which I think we will go over later. Alright, first we're going to go over interphase. Interphase is a very active phase of the cell cycle with many processes occurring in the nucleus and cytoplasm. Interphase basically gets the cell ready for cell division. There's three phases in interphase. The G1 phase, in which a rapid rate of metabolic reactions and protein synthesis occur. The S phase, in which DNA in the nucleus is replicated. So DNA synthesis occurs in the S phase. In the G2 phase, a number of organelles increase, and DNA condenses into chromosomes and spindle microtubules begin to form, basically getting the cell ready for cell division, because the number of organelles must increase because if a cell is divided in two, there must be abundant amount of organelles in both cells, not just one. And for cells that don't divide, there is this phase called the G0 phase. Some cells don't divide and instead they enter this phase. An example of these type of cells are nerve cells and muscle cells. Right here is the diagram we saw in the earlier slide. If you guys want to pause this video now and match up the G1 and S and G2 phase to the diagram, feel free to do so. Alright. Next, we're going to go over cyclins. Cyclins control the cell as it goes through the cell cycle, ensuring that cells divide only when new cells are needed. Cyclins make sure tasks are performed at the correct time. And they also make sure the cell only moves on to the next stage of the cycle when appropriate. For instance, if I don't need that many white blood cells, cyclins would control the multiplication of white blood cells in my body so that I don't have too many. Alright, so how do cyclins work? Well, first, the cyclins bind to enzymes called CDKs, cyclin-dependent kinases, like that. And then CDKs become active and attach phosphate groups to other proteins in the cell. The attachment of phosphates triggers proteins to become active and carry out specific tasks specific to phase of the cell cycle. So basically, there's four main types of cyclins. Cyclin D triggers cells to move from G0 to G1 and from G2 to the S phase. And cyclin E prepares the cell for cell division in S phase. And cyclin A activates DNA replication inside the nucleus in S phase. Cyclin B promotes tasks in the cytoplasm required for mitosis. You guys probably won't need to know the function of each cyclins. However, you guys will need to know how to analyze graphs and data that have information about each of the cyclins. All right, so when cyclin levels reach a threshold concentration, the cell processes to the next stage of the cell cycle. And we already went over how cyclins function in the previous slide. So when a lot of functions, or when a lot of cyclins carry out the function, the process in which we saw in the previous slide, the cell progresses to the next stage of the cell cycle. All right, the discovery of cyclins. Scientists Tim Hunt and Joan Ruderman discovered cyclins while studying protein synthesis in sea urchin eggs. This illustrates that scientists must always be observant, for unexpected discoveries can always be made. This Tim Hunt and Joan Ruderman weren't looking for cyclins. They accidentally found it. This is called a serendipity. It's common in science. That's all I need to know. It's a nature of science question, and you guys probably know that nature of science questions aren't really in the exam that often, but make sure you guys know what to write if it ever is on it. So now we're going to go over mitosis, which is the bulk of this unit. Mitosis is the division of the nucleus into two genetically identical daughter nuclei. And as I said earlier, it's the division of the nucleus, not cytoplasms. The division of the cytoplasm we call cytokinesis. There's 4 phases to mitosis: prophase, metaphase, anaphase, and telophase. You guys probably learned this in middle school because I remember I did, but we will go over it briefly. So prophase is when the chromatin is supercoiled, centrosomes move to opposite poles of cell, and spindle microtubules start to grow from centrosomes. The cell right here, my pointer is so small. There we go. This cell right here is a cell in prophase. And also in prophase, I forgot to mention, spindles attached to chromosomes and the nuclear envelope breaks down. Sorry. So that is a cell in prophase. These cells right here, they're all in interphase. And the way you distinguish cells in prophase and interphase is that cells in prophase kind of like have the shape of chromosomes already, if that makes sense. Anyways, metaphase. Metaphase looks like this. So basically what happens in metaphase is that chromosomes line up at the equator, centrosomes go to opposite poles, and now we're at anaphase after metaphase. So that cell right here is in anaphase. In anaphase, the chromosomes are pulled apart by spindle tubules. So the chromosomes split. Spindle microtubules pull the genetically identical chromatids to opposite poles. Each pole has an identical set of chromosomes, and now this cell is kind of split, but it has to go through telophase, which looks like that. So it basically is the further pulling apart of each chromatid. So the nuclear membrane reforms around the chromosome. So this cell used to be 1, now it's divided into 2 chromosomes, and now the nuclear envelope will form, which will in turn form 2 nuclei. The spindle disappears and chromosomes uncoil and are no longer visible. All right, we're gonna go over the mitotic index, which is defined as the number of cells undergoing mitosis over the number of total cells. This basically measures the rate of mitosis. It's a tool to predict the response of cancer cells to chemotherapy in the hospital. A higher mitotic index, as I said, indicate a rapid proliferation of cells. If you guys see here, you guys could count how many cells are in mitosis and try to figure out the mitotic index if you want. So basically, all these cells that look pretty solid, they're all in interphase. As I mentioned, these cells oh no, not that one, that cell, that one, and that one. And are there any in telophase? I don't think so. So there would be 4 cells in mitosis, and the rest are in interphase. So you would just do 4 divided by whatever the number of total cells is, multiplied by 100, and that would be the mitotic index. All right, now we're going to learn about the condensation of chromosomes. Chromosomes, which are only in eukaryotes, condense by supercoiling during mitosis. For mitosis to occur, chromosomes must supercoil. Alright, so chromosomes are extremely long. I don't remember how long they were, but they were really long, like 2 meters, I think, but I'm not sure. So they must be packaged into shorter structures. During prophase, chromosomes condense, which means that they become shorter and fatter through supercoiling, which means the repeated coiling of the DNA. Chromatin it refers to the state in which when chromosomes are loose and are thread-like. So chromatins are basically not supercoiled chromosomes. A nucleosome is a structure composed of DNA wrapped around histone proteins. Nucleosomes help supercoil chromosomes. That's how they look like. So basically they have an 8 histone core. So 8 histone proteins make up the core, and there's an 1 H1 histone protein that is attached for stability. The DNA wraps around twice around the nucleosome, and these nucleosomes pull together to supercoil chromosomes. All right, cytokinesis. In animal cells, the inward pinching of the cell membrane forms a cleavage furrow separating the 2 daughter cells. This is cytokinesis in animal cells. Cytokinesis in plant cells, however, carbonate-rich vesicles form a row at the equator of the cell and fuse together, forming a cell plate, which fuses with the cell wall, which ultimately divides the cell. And taken directly from the IB booklet, cytokinesis occurs after mitosis and is different in plant and animal cells. Yeah. The cytoplasm divides, resulting in 2 genetically identical daughter cells. And as I said earlier, a little too fast, that cytokinesis in animal cells is this, cytokinesis in plant cells is this. Basically, cytokinesis in plant cells is a little more complex because there is also the cell wall that must be divided. But make sure you guys know both cytokinesises. Alright, so tumor formation would be the next thing we're going to go over. So tumors, if you guys don't know, are abnormal groups of cells that develop in tissues due to cells dividing uncontrollably fast. And some tumors result in cancer. So primary tumors are the original site of tumor. They're often benign, which means that they're not dangerous to humans. Alright, so benign, I defined it, cells remain attached to each other and they don't spread, which means that they're not likely to cause harm. Malignant tumors, however, mean that cells migrate around the body, developing into secondary tumors, which are life-threatening, like cancer-causing. Alright, secondary tumors are tumors formed following metastasis, the movement of cells from a primary tumor. Metastasis is what? The movement of cells from a primary tumor. Alright, oncogenes are genes that can become cancer-causing after mutating. They're usually involved in the control of the cell cycle and division. A mutation, if you guys didn't know, is a random change in the base sequence of a gene. It's triggered by mutagens, which is outside agents that may cause mutations. For example, there would be cigarette smoke, X-rays, and UV radiation. Alright, several mutations must occur for a cell to become cancerous. And if you guys see, the probability of a mutation occurring is really low, but mutations occur quite often in our body because we have billions of base pairs, and even though the probability of of a mutation occurring is low. Since we have so many base pairs, mutations happen all the time, and if we're unlucky, it causes cancer. So, if you guys didn't know, and you guys probably did know, because smoking does cause cancer. But first, we're going to go over what correlation is versus causation. Correlation is a relationship between two variables. There is a positive correlation between cigarette smoking and the death rate due to cancer. This necessarily doesn't mean that smoking causes cancer, but it does. But I'll go over that later. Basically, correlation is not causation. You guys must know that. However, we have enough laboratory evidence to establish that cigarette smoke causes lung cancer. So chemically, we have the data that it shows that cigarette smoke actually causes lung cancer. But for example, let's say I collected data and said all smokers, or 80% of smokers, have lung cancer. However, only 30% of non-smokers have lung cancer. This does not indicate a causation relationship. It only indicates a correlation relationship, and you guys need to be able to distinguish between those two. Alright, that's it for today. If you guys have any questions, you guys can comment it down below, and make sure to subscribe for more content. Thank you.